Then I realized I have to finish writing your test on the weekend. Yay. Okay, I'm not ready for the weekend. Yeah, and I still have to put up your last video for the last lecture, which I'll do like together with this one here. And also your quiz for the week, right? So yeah, it's a lot of work. So things are gonna go up between today and tomorrow, okay? All right, so for today, we are going to do some clarifications on things that I just assumed that they were common knowledge, and then I look back at it and I'm like, I don't think I'm making sense here. Okay, so I'm going to mention a few things that I just brushed through last time, and they were not very clear. Okay, so when I put this slide up last time, I said that those are cell adhesion molecules, and then I didn't really talk about them. And how many of you had cell biology? Okay, so everybody knows what is cell adhesion molecule, so I don't have to go to a lot of details about it, right? Okay, so just to refresh your mind, what are they, right? So those are cells that are, those are molecules that are on surface of cells, and they're very important for interaction between the cell and a different cell, or between the cell and whatever is outside of the cell, like the extracellular matrix, okay? So what is it that these molecules are doing? Okay, so first they are going to allow cell-to-cell interaction or interaction with the matrix, okay? Now, how are these molecules produced? So some of these molecules are always produced from within the cell, so you can call those constitutively expressed, and then some other molecules are induced, and that is very much the case with immune cells. Okay, so what is happening here is that some kind of signal or exposure to some type of pathogen, which in this case here is some kind of fungus, okay? Exposure to some type of pathogen is going to trigger the expression of that adhesion molecule on the surface of the cell, okay? So some molecules are always there, but then some other molecules are only going to come up when you have a trigger for them. Okay, so keep that in the back of your mind. Okay. Now, what kind of interactions these molecules have with whatever they interact with, right? So some of these molecules can interact in a way that they recognize the same type of molecule. So those are type of homophilic interactions. But then some other ones are going to interact with things that are totally different from them. So those are the types of heterophilic interactions. Okay, so this is the big stroke paintbrush version of what is an adhesion molecule. Okay? And then we know that we have 5 main families of adhesion molecules, right? Now, when you say cell adhesion molecule, it gets, the acronym CAMs. Okay, so 5 main families of CAMs. So here they are. You have cadherins, you have immunoglobulin superfamily. Now, usually when you think immunoglobulin, you are thinking antibodies, but those are not the only proteins that have that kind of domain. The immunoglobulin domain can be in different types of proteins, including in cell adhesion molecules, okay? So immunoglobulin superfamily is another family. Then you have the mucin-like CAMs. Then you're going to have the selectins, and then you're going to have the integrins. So 5 families of these sticky molecules. All right. So those are new slides that I put here just to clarify what those families are implying. Okay, what is the role of those families? Okay, so start looking here at the cadherins. So the name of this thing starts with Ka. Okay, so when you see cadherins, so adherins, cadherins. So that is telling you is adherins that depend on calcium. Okay, so those If you don't have calcium present, they are not going to stick to anything else. Okay, they can stick to on a homodimer fashion, okay, like recognizing same type of molecule, or they can do a heterodimer. But if you don't have calcium around, then they are not going to work. Okay, so calcium-dependent adhesion molecules. What kind of molecules are them? Are they? They are transmembrane, so they span the membrane. Okay. And where is it that you're going to find them? In different types of tissues. And how are you going to identify which tissues they are? Well, basically you look at the name of this protein and you say here, okay, cadherin is calcium-dependent adherens. If it has an E in front of it, that means it's from epithelial cells. If it has N in front of it, that is neural cells. Okay, it could also be some endothelial cells. If it has a P, then it's placental, etc. Okay, so the letter in front of the cadherin name is usually going to tell you what kind of tissue this adherent molecule is going to be found. Okay. All right, so let's see here. What are the functions of those cadherins? So basically, they are going to dictate migration of lymphocytes, signaling through lymphocytes, as you can see here. So example of transmembrane signaling, right? And it's going to be very, very important for immune cell interaction with whatever it has to interact with, okay? All right, so let me see here what is important here. So you can read this on your own. Okay. So here, when you look at E-cadherin, that is also important to stabilize interaction between the leukocytes themselves. Like when you have antigen-presenting cell that is interacting with a T cell, we're going to see that at the site of interaction, remember you have to have at least two signals, right? At the site where you're having these two signals, that's where you're also going to have cadherin interaction. And that is going to stabilize the formation of that synapse, that is a space between the two cells, that is doing all this signaling, okay? So the main idea here of the organization of these lectures is really to start from the very basic idea of what's going on, and then you build up on it. Okay, so you always have to keep in the back of your mind, you have your innate immune cells, they're starting to work, they're going to communicate with your adaptive immune cells, and this communication is very, very important, but it's very complex. So we are building on that initial idea, right? Innate cell communicating with adaptive cell communicating with other adaptive cells, and how is it that they're going to start understanding each other, and how is it that they are going to get to the places where they have to go. Okay, so this big picture, we start building up and putting more details and more details in the middle of that concept, okay? So next superfamily here, you have your immunoglobulin domain CAMs. Okay, so those do not depend on calcium. Okay, calcium independent, and of course they're important for, well, immune signaling and cell adhesion, immune response. Okay. Okay, so let me see what is important here. Major types of those immunoglobulin domains. Now you have the ICAMs. First thing that should come to your mind when you see ICAM is extravasation, right? Coordination of the cell going to a specific point where it's going to go through that endothelial layer and get to the site outside of the blood vessel. Okay, so this is intercellular adhesion molecule. And then you also have the VCAMs. Those are more specific for vascular tissue. Okay. Again, the same types of functions. Then you have the PCAM. This guy I have not mentioned before. So this guy is important for platelet type of interaction with endothelial cells. So it's platelet endothelial cell adhesion molecule. What it's going to do is going to, again, coordinate trafficking of those immune cells, but you're also going to have recruitment of platelets. Okay. Okay, so let me see what is important here. Okay. Regulation, they are important for the immune system. Okay, and then you got to these other cool guys here. Mucin-like CAMs. When you see mucin, you think mucus. Okay, when you think of mucus, you should be thinking about polysaccharides on the surface of mucus, and especially polysaccharides that are charged, negatively charged, because those are going to attract water. Okay, so polysaccharides. So those proteins are very much full of sugars. They are heavily glycosylated. Okay, do they depend on calcium? No, they don't. They do not depend on calcium for adhesion. Okay, all right. Now, what is their role in the immune system? They help very much to help those leukocytes to roll on the surface of the blood vessel. Remember when you do the rolling thing before you do the extravasation, right? Now, this whole idea of rolling before you go through is a concept that sometimes you just like brush through it, you know, but if you really think about it, you can have very good analogies with regular traffic. Okay, so let's say, for example, you're driving on 95, right? So if you are driving in the left lane, that is like far away from the exit point, what is your speed usually? Considering you're not on the phone or distracted? Faster than if you are going to get out, right? That's the whole point. If you go in the left lane, and then if somebody is slow in front of you, it's like, this guy's totally out of place, right? And some people really get annoyed, like, you know, okay, you should be going faster if you're away from exit points, right? So in the middle of the blood vessel, that is where you're going to have your faster flow. Now, if you are ready to come out of 95, what is it that you start to do? If you want to come to the exit, you should slow down, right? I mean, if you start to speed up, that is going to cause some issues. I mean, I've seen it, but you know, you guys like all the way on the left. Oh, my ass is green, and then what can happen? A crash or several crashes. And yes, you can put a good analogy with your blood vessels, because if you have a very fast blood cell going from the middle and the cell has to get out and decide to stop and go, they're going to crash. And that's exactly what's going to happen, because the flow close to the blood vessel, close to the endothelium, is slow. If a fast cell gets in there and the other cell trying to get out suddenly just stops, they crash and they burst. Okay? So the whole idea of getting close to the endothelial layer on your blood vessel and slowing that traffic, slowing that flow, is really, really important to first keep a laminar blood flow, means without turbulence, right? And the second, to make sure that the cells are first slowing down without crashing each other, and then they'll be able to adhere, strongly adhere and do the extravasation. Okay, so you want to create the least amount of turbulence or crashes with those cells. Okay, so when you look here at those ICAM molecules, they are really important for that rolling motion of those cells. So they're not going to just stick like the Velcro or crazy glue. They're going to roll and kind of weak interactions. Okay. So an example here is your P-selectin, and that is going to bind to the E-selectin on the endothelial cells. So E for endothelial cells. Okay. And that's, you know, that's really causing your leukocyte rolling. So trafficking and this rolling, this is a very important function of those mucin-like cells, CAMs. Okay. Another function is T-cell activation and homing. Okay. So what that means is that those glycoproteins are also going to allow interaction of leukocytes to each other and adherence, so you can have cell-to-cell signaling. Okay. Right. Now, the homing part is what we're going to see next now that, was for the last class, but is going to be more explained now. The homing part has to do with interaction of those lymphocytes to specific places where they have to go. Okay, so keep in mind, this is going to be like a zip code that those cells are going to get. Okay. All right. And then you have your selectins. Right. So selectins are also transient interactions, so they're not like sticking like crazy glue. Okay. Now selectins, they also depend on the presence of calcium for them to bind to their targets. Okay. So selectins are going to interact with some kind of carbohydrates. Okay. So with calcium, they interact with some carbohydrates, and again, this is always on cell-to-cell signaling or cell interaction with the environment. So we do have all different types of selectins, like L-selectin. The L is for leukocytes. You have E-selectin for your endothelial tissues. Then you have P-selectin for platelets, but also for endothelial cells. Okay, and of course, to make things more complicated, they get cluster of differentiation number, but we're not even getting into that. All right. And then the last type here is your integrin family. So integrins are by nature heterodimeric. That means they are made up of two parts. Okay, they cross the membrane and they're made up of two parts. So here is your cell membrane. Okay, so what are these two parts? You have an alpha and a beta part. Okay, now looking over here, you see, okay, so they have alpha. Let me just highlight here. Alpha part, and you have a beta part. Okay, so beta subunit, alpha subunit. So those integrins on the surface of the cell, they can be in one of two conformations. They can be either in a low affinity or inactive form, or they can be in the active or high affinity form. So this one that I had drawn over there is actually the active form. So over here, I'll draw the inactive form of those integrins. They're going to be something like this, like they are tired, they are laying their heads on each other, like, oh no, yeah, let's just go to sleep here. Like sometimes when you are, I was going to say on a bus, but nobody takes the bus here. So let's say if you're on a plane, right, on a long flight, an overnight flight to somewhere else. Anybody flew out of the country, like more than 8 hours overnight flight? Yeah, just a few people. Yeah. Where'd you go? Italy. Oh, that's my next target. Yeah. So Italy, Europe, somewhere else, But anyway, it's just a brief story. I flew to India. Okay. There was a long flight, yeah. And for some reason, some people, I don't know, they don't like to, I think there's something with the culture, they don't like to use a lot of deodorant, but anyway, there was this guy sitting close to me, and the guy was so tired, and the head was always on my shoulder. For the first 5 hours, I was like, kind of like move, but after 8 hours, the guy was so exhausted. I was thinking, as long as you don't drool on me, fine. But anyway, that's the inactive form, kind of like comatose. But anyway, so this thing can have two conformations. It can be inactive or it can be active, right? So when it's laying down, it's kind of the inactive form. What is the integrin important for? It's very important for trafficking of those leukocytes. It's important for antigen recognition, and is important for the response to the antigen recognition. Okay, so important for a lot of aspects here. Now, look at this. Important for migration, adhesion and migration, right, extravasation. And here I had mentioned before one protein that was LFA1 binding to the guy ICAM1. So when you see ICAM1, you think extravasation, right? So the integrin that binds to ICAM1, that integrin is called LFA1, okay? So that's important for sterilization. And the ICAM1 is on the endothelial cells of your blood vessels, all right? When you get to the point of interaction of LFA1 to ICAM1, then you get to the strong interaction. That's what is going to push for the extravasation, right? So this LFA1 has to be at the conformation that is going to allow the interaction. There is going to be a high affinity type of interaction. Because if that LFA1 is on the dormant state or low affinity, then it's not going to extravasate. Okay, so it needs to get a signal that is going to tell it to be in the right conformation in order for the cell to come out of the blood vessel. Okay. Okay, so different integrins and different types of functions that they have. This is FYI, don't go crazy memorizing, okay, for your information only. I'm not going to test you into this. I'm going to only test you on the overall picture and overall functions of integrins, okay. But you can see here that when you have here this guy, LFA1, which I mentioned before, you're going to find those in white blood cells. And they are important for adhesion, okay, and T-cell activation, and also, of course, for sterilization. And then you look at other types of integrins, you're going to find some in innate immune cells, like macrophages and neutrophils, right? And yeah, so basically you find them in all types of cells, and they're important for all types of responses. But you got to remember that they are in this heterodimeric form, and they can either be sleeping or they can be awake. All right. Okay, so here is the first question. Yeah, I did have this question on the first slide, but then I thought that would be kind of mean to people who would walk in late in the class, right? So I just push it down. So here, this slide is still referring to things we had talked before, right, about the cytokines and how your cells go towards a place where they have to come out of circulation, right? How do they get to the place where they are needed? The question here is, which of the following best describes the migration of lymphocytes via the chemokine receptors? So what is attracting those lymphocytes to a specific area? Okay, anybody need more time? Oh, okay, so what is your answer here? We'll go for B. Okay, so remember the chemokine receptors are mainly G protein coupled, right? And those lymphocytes are going to travel up the chemokine gradient to via the interaction of the chemokine with their G protein coupled receptors. Okay. And I was looking here at D. It looks kind of interesting too, but I did not mention enzyme link chemokine receptors. Anyway, your answer here is B. Okay. All right, so here is the story, continuing the story of those cell adhesion molecules, right? So this is your integrin guy, integrin story. So what kind of conformation is this here? Is it very high affinity or is it low affinity for binding stuff? is low affinity. The guy is sleeping on somebody else's shoulder, right? The binding site is away from interacting with anything else, okay? So at first, you have this low affinity, and this integrin is quiet. Now, there are two ways in which you can activate this type of integrins, okay? So one way has to do with a signal that is coming from inside of the cell, okay? And then you should ask me, how is that signal going to get inside of the cell in the first place? Oh, well, something else interacted with another receptor, sent a signal to the inside of the cell, and the inside of the cell is started to produce this protein that is called tallin. And then tallin is going to interact with the cytoskeleton of the cell that is mainly made up of actin filaments, okay? And then interacting with the cytoskeleton, this tallin is going to get close to the cell membrane. And when it gets close to the cell membrane, it will be interacting with the cytoplasmic side of that heterodimer molecule. When it touches the cytoplasmic side of the heterodimer, it is going to send a signal that is going to wake up this dimer, okay? So the dimer is asleep, and then tallin goes there and bites this guy's foot, so it wakes up, okay? So now they can be able to interact with something else that is outside of the cell. So we got a signal from the inside of the cell that is going to interact with something on the outside of the cell. So that is called your inside-out signaling, okay? All right. The other type of signaling that you can get is the outside-in signaling. So what happens here is that this dimer here, most of the time, it is in the low affinity if it's not bound, right? If it's not bound to ligand. But sometimes, or a very small percentage of the time, this protein is going to shift and stay a little bit by itself in the high binding conformation. Okay. If you have a lot of ligand around that is ready to interact with the high binding state, then the ligand is going to bind and is going to lock this conformation state of the molecule. Okay. So once you have one of those cells bound like this, one of those molecules bound like that in your high affinity state, you can have other molecules of the same type that can also bind to ligand and stay in that conformational state. Okay, so when a bunch of those molecules get together, they're going to find a cluster of high affinity integrins in this particular area, and that cluster is going to allow intracellular signaling of those domains that are ready to interact with something else. Okay, so this type of signaling really has to do with clustering of those integrins. Okay, this is why you have those brackets here. So you need like n number of those molecules to be interacting in order for the cytoplasmic domain to send a signal to the inside of the cell. Okay, so that is your outside in type of signaling. And here again, you have here the important things that are highlighted and bold and everything. Everybody cool with that? Inside out, outside in. Okay, all right. Okay. So here's another question, which is an easy one for now. Which of the following proteins activates the integrins by linking them to the actin cytoskeleton? Okay, everybody has to get this one right. Unless you are in your inactive state, right? Does anybody need more time? No? Everybody submitted the answer already? Yeah, I'm going to stop it. What's your answer? See, that's your talent. All right. Now, fun part starts. Okay, so trafficking of those guys to mucosal tissues. Why do we want even to talk about this, right? Anybody? How do you get some kind of infection? Two ways, right? Is it going to be either through mucosal tissue or is it going to go through your skin? How else is something going to get inside of your body, right? So when you start talking about lymphocytes, they're going to help you to fight the infection. You really have to know where they're coming from, and they have to know where they're going. Okay, so let's see what is happening here. This is representing your gut. Okay, so let's say here you have invasion of this bacteria that went through your gut and ended up in this secondary lymphoid tissue that is called a Peyer's patch. Okay, and in there is going to start activating some T cells. Okay, so what's going to happen here? Your T cell start to get activated. It picked up something, or it could be also innate immune cell, like for example dendritic cell, okay, that pick up the pathogen. Then these things, they're going to travel to the first lymph node that they can find. When you're talking about mucosal tissue, the lymph nodes, they are not called just lymph nodes. They are called mesenteric lymph nodes. Okay, so this, let's say here, this T cell travel to the first lymph node that is a mesenteric lymph node, and here is going to get activated, right? It is going to find a B cell to activate, and then let's see what happens here. And then it is going to come out of there, and if it doesn't go back to the site of inflammation or the site of infection, it's basically useless. Because if you have an activated cell that stays in the lymph node, it's what you're going to do with it, right? You have nothing to do with it because your infection is over there. And then you have a further problem to that infection because this is your gut. What happens in your gut? Absorption of nutrients, yeah, and what happens in the gut doesn't usually stay in the gut, right? So it starts over here, and then you have peristalsis, and this stuff is moving down. So the bacteria that was started over here, it can end up over there somewhere in the lower part of your gut, right? If you're lucky enough, it will come out of your gut, but most likely it's going to just infect other places along your gut, okay? So now what's going to happen here? What is the whole rationale of this? So you get a cell that went to your meeting place here, mesenteric lymph node. It got activated, become an effector cell. So now it's going to come out of your lymph node through these efferent lymphatic vessels. It's going to be dumped in the blood circulation. Okay, where is it dumped in the blood circulation? Close to the vena cava, all the way up here somewhere, okay? It goes to the right atrium and the heart pumps it all around. So now it's in the blood circulation. How is it going to know that it has to go back to the gut? That's a problem, right? Because it can go everywhere in the body, but it is needed in your gut. Not only is needed at the site where you first found the infection, it is needed all along the gut because your infection can be down there right now. How are you going to know how to find this thing and kill it? Okay, so this is what happens here. Okay, the idea of homing of those lymphocytes to the place of infection, that is this really cool zip code type of idea. Okay, so you're going to have something in the gut that is going to interact with something else in your lymphocyte, and it's going to tell you exactly where to go and where it's needed. Okay, so keep that in the back of your mind. So that's with the gut. And here is also another place where you can get infected, which is through your skin. Okay, And the same story happens here. So this is representing the top layer of your skin, which is the epidermis. Okay. Everybody took AMP1, I assume. No, or AMP2. I don't even know where I taught this thing. One of the AMPs. Okay. So you have your skin. The outer part is your epidermis. The very much outer part is dead cells, right? But then you have this thin layer here that doesn't have any blood vessels. Okay, so let's see here. In your epidermis, there are no blood vessels. Okay? So if you don't have blood vessels over there, it's like, how the cell's going to get there if it is in the blood circulation, right? Well, when you look at the dermis, you're going to see that the dermis has blood vessels. Yes, blood vessels. Okay. So you look at the population of immune cells that are present in both dermis and epidermis, you're going to see they're quite diverse. So if you look at the epidermis, you're going to find dendritic cells. Okay. If the dendritic cell is just sitting over there in the epidermis, it usually has this other strange name that is called a Langerhans cell. But in latest textbooks, they just call dendritic cells, like resident dendritic cells. Okay. Besides those dendritic cells, you're also going to have some specialized form of T cells. Okay, there we're going to see, how they differentiate. You're going to have the T cells that we have seen before. Those have the receptors. They are alpha-beta receptor. And that you should remember how the beta goes in first and then the alpha. That's the alpha-beta receptor. Now, besides that, you're also going to have this strange-looking T cell that has a different type of receptor. That's called gamma delta. Those T cells are a lot more aggressive than your alpha-beta T cells. They can be triggered by basically anything. Okay? And they have a special superpower that the alpha-beta T cell doesn't have. Okay, so if I ask you, how is it that the alpha-beta T cell sees antigen, you should say that they see a little stretch of protein that is presented on MHC molecule, right? Now these guys here, these gamma delta T cells, they don't need that. They can see 3D. They can see just like, not just like, but similarly to B cells. And they are placed strategically in areas that are more prominent to getting infected. Okay, so they are placed in sites where if something comes across, they will feel it, and if it's not self, they will kill it, and they won't even ask questions. Okay, that's the type of cell that's like, I'll kill you first, I'll ask questions later. Right? Anything that comes across they don't recognize as good, it's bad. Okay, simple-minded, the type of T cell. All right, so these cells are sitting around close to the skin, right, the epidermis part. But then when you go deeper, you're going to see that you have more types of cells that are going to be hanging around there. And then these cells over here, they are able to come from the blood vessels on this side here, okay, the dermis side. Once they are in the dermis, then they can migrate to the epidermis, but in order to get there, they have to come from the blood vessels. Once they are out of the blood vessels, they will have, if they want to get back into circulation, and if you want to get back and go to lymph nodes, then they have to go through the lymphatic, right? They have to go through the lymphatic circulation. All right, so those are areas where you want to find cells that are going to fight infections, okay? So the whole idea here is, how is it that your lymphocyte is going to get to the place where it has to fight infection, right? Remember, it gets blood dumped into the blood vessels, into the blood in general, and then the blood is flowing through blood vessels. Now, this poor lymphocyte that is here inside of the blood vessels, it cannot see anything that is outside of the blood vessel. It has no idea what's out there, okay? So now getting again here the analogy of you driving on 95, how do you know where to get out of 95? Let's say you want to come here to FAU, right? How do you know which exit to take? Signs, right? You have signs. And you say, okay, this is close to Glades Road. That's where I have to go. Guess how these guys get to the place where they have to go? They use signs. Okay, so once they got activated, once they went to the secondary lymphoid organ and they got activated, they have the receptors for specific signs. Those receptors, they're upregulated because of the initial message that they got from the dendritic cell. Okay, so the dendritic cell is like, okay, I have seen a worm in the gut. Okay, so the dendritic cell goes to the lymph node and is going to tell the T cell, like, look, there is a worm out there in the gut. So you're going to have to upregulate certain type of receptors because you're going to have to figure out where the gut is, and you're needed in the gut. Not only at the place where I found the worm, but everywhere in the gut, because the worm can go down, can go up, you know, it can go everywhere. Okay, not only the worm can go up and down in your mucosa or in the gut, but then the worm can spill over and go to your lungs too. So just in case, just go to the lungs too. Okay, so this is all the message that your T cell is getting from your dendritic cell. Okay, then what's going to happen here? Look. Here, huh?
This is the T gamma delta, or just a regular T cell?
This is regular T cell. Regular. Okay, so your vasculature, your blood vessels are going to have the addressins. Those are your cell adhering molecules, right? Those are your CAMs. They're specific to the blood vessel at a particular place. Okay, so it's really like a zip code. Okay, so let's see here. Okay, if you have, for example, here molecules that are this type of molecules, okay, so you're going to have a receptor that is, or enough receptors that are going to be able to interact with all of those types of molecules that are present in the blood vessel. Okay, so those co-receptors are going to be upregulated in your lymphocyte, and it's really going to be like a zip code. So let's say if you have at your destination this like 1, 2, 3, 4, 5 type of molecules, then you are going to have upregulated like the 5 type of receptors for those molecules. Okay, if you have only 2 or 3, you're most likely not going to get to the right place. Okay, so let's see what happens here. If your thing is intended to go to the gut, so you're going to have molecules here in the blood vessel that go to the gut, and those molecules are going to be like L-selectin, VLA4, LFA1, and this type of interactions of molecules. Okay, so that's specific for gut zip code. If instead you want to go to the lungs, then the interactions are going to be different. You're going to have different kinds of interactions that are going to be needed to send this cell to the lungs, and so on and so forth. So every tissue is going to have a specific type of protein expression in the blood vessel that is going to tell your lymphocyte that this is the place where it has to come out of the circulation. Okay? So it goes to the zip code, and now here you're getting a lot of interaction. You're getting rolling, and you're getting stopping, and then you're going to go through extravasation, because really, I mean, from the inside of the blood vessel, everything looks the same, right? So if you don't have those signs, you don't really know where to stop. Okay, so every destination or every organ is going to have a specific set of molecules that is really going to tell your lymphocyte where it is needed. Does that make sense? Yeah, zip code, right? Okay. So here, when you look at your T cell, it's going to have the receptors. So here is an example of T cells that were not activated. They only have the basic receptors. Okay, so if you look over here, like we have seen before, a naive T cell is always going to have this receptor for CCR7, because that is a sign for the T cell to keep on recirculating, keep on patrolling. Okay, this CCR7 is going to tell the T cell that it has to go inside of your secondary lymphoid organs. It has to go to the lymph nodes. It has to hop from lymph node to lymph node. It goes in series, right? Goes back to the blood circulation. It checks out the Peyer's patches. It goes back to the blood circulation. It's totally naive. It keeps on recirculating, right? Now, if your T cell is not naive anymore, it is called an effector T cell. Okay? It can be either effector or memory T cell, but now let's say that is an effector T cell. An effector T cell is a trained soldier that has to go to battle and do what? Kill the pathogen, and then I did bring chocolate. Somebody from the back. Anybody awake back there? That's part of the killing the pathogen. Okay, you gotta. Almost. All right, yeah, is the reactions going on, and then what's going to happen to the T cell? Got more chocolate. Take a guess. Okay, just analogy with the Braveheart guy, right? What happened to the soldiers? They're gonna die. Want a chocolate? Right? Oh, almost forgot. Oh, perfect. Okay. They're gonna die. Did you tell her? Ah, that's not. Okay, give me chocolate. She was in my office asking these questions. I told her, okay, so your soldiers are gonna go to battle, and they're not supposed to come back to your secondary lymphoid tissues because you're getting trained as a secondary lymphoid tissues, and those guys are already trained. They're ready for battle. They're gonna go, they're gonna kill the enemy, and they're gonna die. It's a cruel world out there, right? So what's gonna happen here? Those guys, they're effector T cells. They don't have CCR7 receptor anymore because they're not supposed to go back home. They're supposed to go out and die after they do their jobs. Okay, so look at the difference here. No CCR7 for effector cells or for memory cells that are in the periphery. Now this is not a complete chart, of course, because if you have memory cells that stay hanging around in your lymph nodes, those have CCR7 receptors because that's what brings them to the lymph nodes. Okay, so you have memory cells that are out there without CCR7, and you also have memory cells that stay around in the lymph node, and those do have CCR7. So this is an important concept to have. Okay. All right, let's see what else is important here. This is just the interaction that those receptors are having. Okay, remember CCR7 is interacting with CCL19/CCL21, right? I don't want to look like it's not good. Okay, and those, where do you find those guys? Remember the name of those specialized vein side of capillary beds? What do you call them? They're very tall. HIV? No. Yeah, high endothelial venues. CCL19 and CCL21, those are trademarks of your high endothelial venues. That's the zip code for your cell that there is a lymph node right behind it. Okay? So this is the idea of homing and using those zip codes to direct the traffic of those T cells. Okay? Oh, more question. All right. What are vascular addressants? There's only one answer that makes sense here. Anybody need more time? Okay. Okay, 5 seconds. Okay, what's your answer? It is D. Ligands to the homing receptors of the lymphocytes. That's the whole idea of those addressing. They're giving you the address of the thing that is right behind your blood vessel. So this is vascular, that means your blood vessel, right? What is the address of the blood vessel? So those are ligands to the homing receptors on your lymphocytes. Okay, everybody cool? All right. So here. So now we're going to get you more details on how this interaction is taking place. How is it that you're going to see the antigen and what is it that you're going to do with it, okay? So remember, you have this eating cell over here. The here is exemplified as a macrophage, but it could be a dendritic cell, most likely, right? The dendritic cell. So, sorry, those antigen presenting cells, they have on their surface, they have this molecule that is MHC class 2, okay? And once they process the antigen, they cut a piece of it and they put on the top of that MHC class 2, and they'll be ready to present it to a T cell. What kind of T cell? Helper T cell. Helper T cell has CD4. CD4 is going to interact with MHC class 2. Okay, so here is your helper T cell that is going to recognize MHC class 2, and then what's going to happen to the helper T cell? Well, first thing is going to help stuff by secreting chemicals like cytokines. Helper T cells are very good at secreting interleukin 2. Okay, they're very good at it. Okay. And then what's going to happen? Yeah, of course, T cell is going to get activated. And after that, they are still going to go around and activate the B cells. Okay? But there's more to the story. All right. So here is the initial story with your dendritic cells. Here is your very good eater dendritic cell. And that's the professional guy who is going to go to the lymph node and present the antigen to your T cell. Okay, that T cell was naive, and the way that the dendritic cell is going to activate your naive T cell is by having that second signal that goes through that B7 and CD28 communication. That's really, very important because the dendritic cells are the ones that have a lot of B7 on their surface. That's why they're very good at activating T cells. Okay. Some other antigen presenting cells don't have as much B7 on the surface, meaning they can't really activate naive T cells. But the other antigen presenting cells can activate effector T cells that are memory T cells, like here. Okay, so this guy here is a effector memory T cell. It doesn't need two signals anymore. Now, with only one signal that is getting from this other guy that is not as professional in presenting antigen, it can just recognize that antigen. It doesn't need a co-stimulation because it is already a effector. Okay, only the naive needs an extra push. Okay, so this guy can go and do his job just with one signal. Okay, and same thing goes here with the presentation from the B cell that can eat up. Also, B cells are phagocytic. Okay, so they can eat up the pathogen and process, put it on the surface. B cells can also activate effector memory T cells. okay? But the ones that are really, really good at activating the naive is the dendritic cell, just because it has that stimulating ligand B7. Okay, very, very important for activating your naive T lymphocyte. All right. So everything here that I just spoke about the dendritic cell. Remember how they work. Okay, they are sitting in the place like interdigitating the dendritic cell. At first they're quiet, they're very hungry, they don't want to be moving around. Bye, see ya. And after they eat something, they are going to start running to the nearest lymph node. They migrate, right? Then they're going to look for the T cell. Okay, the dendritic cell really is the bridge, okay? Bridge to your adaptive cell. Now, the story gets more interesting. Okay, so let's see what happens here. Your T cell, you know, they start out as naive, and then they're going to get activated, right? You know, they need two signals, right? So now looking again at those two signals, first you're going to see what is the first signal is antigen recognition right there. So you know that your antigen is going to be recognized at the hypervariable region of your T cell receptor, right? The beta and alpha chain, okay? But besides that, you also need that second part of your T cell receptor, that is your CD4, that is going to interact with a part of your MHC class 2. So this whole thing here is your first signal, okay? And then of course B7 CD28 is going to be your second signal, okay? Now, in case you have cytotoxic T cells, right? So they have T cell receptor, but here they have MHC class 1. I mean, they have CD8, right? CD8 on their surface that interacts with MHC class 1, right? So the same thing goes to that type of interaction. You're going to have to recognize the antigen, and then you're going to be able to respond to the antigen. But then cytotoxic T cells behave very much differently than your helper T cells. We're going to get there. I don't want to run too much ahead of time here. All right, so here, let's see what happens here. Your T cell receptor, okay, is called a CD3 complex. What makes up that CD3 complex? You're going to have here your T cell receptor, beta and alpha, okay? You're going to have your CD4 molecule or your CD8 molecule, okay? And you're going to have a CD3 molecule that is complexing with alpha chain and beta chain that protrude to the cytoplasm. Remember the alpha chain and beta chain that are important for, Ig alpha and Ig beta. This stuff is almost the same names, but it gets complicated. Alpha beta is part of the receptor. Ig alpha and Ig beta is part of the CD3 complex that sends the signal to the inside of the cell. All these things are part of your T cell receptor complex, okay? Both your cytotoxic T cell as well as your helper T cells, they have these adjacent molecules that are important for signaling, okay? So looking again here at your receptor, you have your beta chain, you have a constant region that goes to the cytoplasm, that goes to the membrane. You have your variable domain that is together with the variable domain of your alpha chain, and then your constant region goes on the other side. It also spans the membrane, right? We see the receptor here. Remember that your T cell receptor is always stuck on the membrane, is never secreted, right? It doesn't work like an antibody, right? Okay. So here, again, the story on what's happening here. You have your receptor, you have your CD4 that is part of your CD3 complex, part of your receptor. CD4 is very important for recognition of your MHC class 2. So when your T cell receptor recognizes the antigen, this thing is coming close to the CD4 that is going to recognize the MHC class 2. Okay? That signal is very important in order to tell the cell to do something once you recognize the antigen. Again, this whole thing here is only signal 1. Okay? When you look at your cytotoxic T cell, it has the same type of setting, but then instead of CD4, it has CD8 on the surface. Then CD8 is going to interact on the side with MHC class 1. We're going to see more on this. Okay? All right, so let's see what happens here. Remember that B7 and CD28, this is your second signal that is going to tell your T cell to get activated, right, your naive T cell. All right, so there is more to that here. When you look at this interaction between your antigen-presenting molecule and your T cell, and then the B7 and the CD28, this whole space in which these two cells are interacting, that is called the immune synapse. Okay, so it's space in between the interaction of those two types of four types of molecules. That is really your immune synapse. All right. Now, what is that going to do? Okay, in the case of a helper T cell, okay, when it gets activated, meaning signal 1 and signal 2, this helper T cell is going to start to produce certain proteins, and specifically is going to start producing a piece of a receptor that it has on the surface. So that receptor is a very important receptor for interleukin-2. Okay? Now, originally, the T cell has only two parts of that interleukin-2 receptor. It has the beta and the gamma part. Now, these pieces of receptor by themselves, they are able to bind to the ligand, which is interleukin-2, but they cannot bind it really well. So it cannot respond to interleukin-2 presence. Okay? The only time the cell is going to be able to respond to interleukin-2 presence is if it gets those two signals, upregulates this third piece of the receptor, and puts it on the surface. Now it will be able to interact with interleukin-2 ligand. Okay? The analogy here is that the receptor is there, but is not very effective at biting the interleukin-2, which in this case is the apple. Okay? So this receptor here is really trying to bite the apple without teeth. You can try, but you won't be able to. Okay? Now, if you get signal 1 and signal 2, now you can produce the teeth on your receptor to bite off a piece of the apple. Okay? So now your receptor is effective. So this guy that got activated not only is going to make its good receptor, but it's also going to produce interleukin-2. This guy is making the ligand that is going to bind to its own receptor. Okay? So this is called an autocrine type of signaling. Okay? Now, since this guy is activating itself, okay, it could be close also to other guys that got activated and could make use of interleukin-2 if they have the proper receptor. Okay, so that type of signaling that is signaling to cells that are close by, that is called paracrine signaling. So if the thing is activating itself, that is autocrine. If the thing is activating a cell that is close by, that is paracrine. Okay. So story with the T cell, first thing that it does, upregulates the receptor that can bite, and then it upregulates the apple, the interleukin-2. What is interleukin-2 going to do? It's going to tell this cell that it has to divide. You have to have a whole army of these guys because this guy recognized the antigen. There was a piece of the pathogen. So you're going to need a whole army to go and kill the pathogen. So interleukin-2 is going to tell the cell to proliferate. Okay, and T helper cells are very good at producing interleukin-2. Okay. All right, so let's see here. So that's the story with initial activation of your T cell, right? So T cell is dividing and is going to start doing its job, which you're going to see next. In order for your whole immune system to be working, of course, you're going to need your B cells to be working too. right? So how is it that the B cell is going to get activated? Right, so let's look at B cell activation here. Now, once the B cells activate, they become plasma cells. That means they're going to start to shoot out antibodies, right? Remember the sequence of the genes that is making the B cell receptor? The first antibody to come up is your IgM. And because of alternative splicing, you can also get IgG, right? So you have IgM and IgG, mainly IgM on the surface of this cell, okay? So now your cell is naive and is ready to get activated. Okay, so let's see what happens here. B cells also are going to need two signals, okay? Not only the signal to the B cell receptor, but it's going to need two signals. Okay, so let's see what happens here. When your B cell is going to get activated, remember where they're sitting in your lymphoid, in your lymph node, is sitting in the B zone, okay? And if it gets activated, then you're going to start making the follicle, so the primary follicle. Well, because if your cell gets activated, you're going to have, you need a whole army of those B cells as well, right? So that thing is going to become a germinal center, and that is your primary follicle, and then your secondary follicle. And this is all happening inside the lymph node, okay? So let's see what happens here. There are two ways. This is a part that gets a bit, you got to put some brain power. So let's wake up, right? Everybody awake? Let's put blood to your brain. Like, yes, I'm awake. Okay, if I don't see arms up, you're asleep. Yes, I'm awake. All right, so everybody awake. This is important, okay? It's a clue. I'm going to ask you about it. Okay, so, okay, so what happens here? There are two ways for you to activate your B cells. One is called T-independent. Guess what the other one is called? T-dependent. Okay, so this, looking at the T-independent first, it means that you don't need T cells. All right, that's easy. But remember, you need at least two signals, right? So how is the cell going to get two signals? Let's see here. Here is your B cell receptor. Remember that is also complex with Ig alpha and Ig beta, right? Besides that, it has another molecule that is characteristic of B cells, is also a marker for B cells, and that is CD19, and the guy is an ITAM. Remember the ITAMs? Yeah, they'll show up again. Okay, so this guy here is an ITAM. Everybody's close here to your immunoglobulin, and this guy is IgM, right? It wasn't activated yet. Now let's say here that your IgM here is specific for a type of protein that is on the surface of a bacterium, okay? So if this bacterium goes in there, and now this immunoglobulin here, this IgM, recognizes that one piece of protein, and is only one of them that recognizes it, that's one signal that means your B cell is going to become a zombie, just like your T cell becomes a zombie if it has only one signal. Okay. What's going to happen here, you're going to have more B cell receptors on the surface of the B cell. They're going to be able to interact with that same antigen. So now, here you're going to have signal 1, and here you're going to have signal 2. So hey, now this cell is self-activating. You got the two signals. Okay, you use a little trick because, you know, it is one molecule here, another molecule there, but okay, you got two signals. All right, so B cells can do that. Okay. Once you activate the B cell, then you're going to send signal transduction to the nucleus, and you're going to start to make inflammatory cytokines. Remember the most important trio of inflammatory cytokines? Well, if you don't, they're right there. Okay? Okay. So, more on the T-independent type of mechanism. So the first way of T-independent is two antibodies. There is another way that it can be activated by using the antibody. And look at this. This is a cool trick. Okay? Remember when we were talking about the complement system? Oh, it will come again. Okay, so this guy here is a piece of complement protein. Remember the C3 guy that was the most important one? So a piece of it is able to non-specifically bind to pathogens. They just stick to pathogens. That's what they do. That's their job. Okay, so when that piece of C3 gets stuck to pathogen and is floating around, if it finds a B cell that recognizes the pathogen, that's good news, right? Now, that pathogen or that piece of pathogen, not the whole pathogen, otherwise it's going to be big, right? That pathogen is coming with the C3 piece of molecule. The C3 part has a receptor on the surface of that B cell. The receptor for the C3D molecule is called CR2, okay? Complement receptor 2, okay? It's also called CD21, but I'm only interested in CR2, okay? So now what's going to happen? Recognizing the pathogen by the IgM is going to be your first signal. Interacting with CR2 is going to be your second signal. So now you're going to activate your B cell. You got two signals. All right? So this is your other T-independent type of signaling. So two ways of T-independent. Okay? And now we're going to look at the T-dependent type of activation. Okay? So what does that say? Remember your B cell, oh, it's missing stuff here. Okay. Your B cell that could eat a pathogen is also phagocytic, right? Your B cell is going to present a piece of that pathogen on MHC class 2. And that is going to interact with your T cell. And the T cell is going to be able to cross-communicate with your B cell, okay? Now, depending on the nature of the pathogen, the T cell already got trained on what to tell the B cell. Okay, so let's say here, for example, your pathogen was a virus. So what was the first thing that happened? The dendritic cell ate the virus, presented to the T cell, and already told the T cell this thing is a virus. Okay, so T cell gets to interact with the B cell, is going to secrete interferon gamma and other cytokines, and is going to tell the B cell to become a plasma cell that produces IgG on the surface. Out of the cell. It's going to kick out IgG. Okay? Some IgG is still going to stay on the surface, but this is going to be a machine gun of IgG that's going to be shooting out of the cell. Okay? So depending on what your T cell tells the B cell, that is going to tell the B cell to do the class switching. The only time you're going to get B cell class switching is when you have activation by the T cell. Okay? If the B cell is only self-activating, you never get class switching. You only get IgM that is secreted, okay, as a pentamer. Okay. Now, once your B cell goes through the class switching and starts to make specific isotypes of antibodies, it can never go back to making IgM. Because once the T cell tells the B cell to switch, the way that it's telling the B cell is by cutting out a piece of the DNA from the B cell. So the B cell is losing the genes to make those early antibodies. It can never go back. Once it starts to make IgG, that's all it does. It's going to make IgG. It can shift to another type of IgG because you have several genes for IgG, okay? But it can never go back to make IgM, okay? Did everybody get that? Hopefully. Okay. Lisa, check if you're awake, right? Okay. So how is it that your T cell, your helper T cell, is telling your B cell to do the class switching? Is by recognizing a common antigen right there, right? So B cell receptor and T cell receptor, this is MHC class 3, okay? MHC class 2 on your B cell interacts with your T cell receptor. That is a signal. And this part here that is really, really important. CD40 receptor on your T cell gets together with CD40 ligand on your T cell. And that is what is going to send the signal to do the class switch. Okay? Class switch of the antibody. All right. Okay, so class switch to what? Okay, so let's say that at first your phagocytic innate immune cells ate up a piece of worm. I always like the worms, okay? So it ate up a piece of the worm. So it told your T cell to produce certain types of cytokines like interleukin-4 and interleukin-5. When the T cell communicates to the B cell, and instead of giving it interferon gamma, it gives it interleukin-4 and interleukin-5, now your B cell is not going to make IgG because IgG is used against viruses. What your body needs is IgE that is going to be used against worms, helminthic worms in your intestines, for example. Okay? So these cytokines, interleukin-4 and interleukin-5, will train your B cell to make IgE. Isn't that cool? So depending on what your T cell has seen at first, it is going to tell your B cell what kind of antibody to make. Okay? Again, here, if your T cell has seen a dendritic cell with a virus, a piece of a virus, then your T cell is going to produce interferon gamma, and that is going to tell your B cell to class switch to IgG, because IgG is really good at fighting viruses. Okay? But again, what is the first type of antibody that a plasma cell will produce? IgM, before any class switching. And that goes for any type of infection. Make a note of that. Okay? It's important. Why? Because when you have an infection and you want to check if your patient already had this infection before, if it is an old infection or if it is a recent infection, you're going to check for levels of antibodies. You're going to check for the presence of IgM and for the presence of IgG if your pathogen is a virus. Okay? If you see high levels of IgM, that means your person got infected recently because your B cells didn't even have time to class switch yet. Okay? Presence of IgM in the serum of a patient who's infected, that is indicative of recent infection. Does that sound like a good test question?
Yeah.
Yeah, I like it. All right, so let me see what is the next one here. Okay, I'll go to this. Okay, this is only the detail on how to do the class switching. It has to do with expression of this cool enzyme that is called AID, and is for activation-induced deaminase. Okay, so cutting a piece of the DNA allows for the class switching. And let me see if I have a lot more to say here. Okay, so I'm going to stop here, and we'll pick up on Tuesday.